Okay, so what we want to do with this video is show you how to do a standard plate count. What I have here is a tube of E. coli. This E. coli was inoculated 20 hours ago. It's filled with bacteria. What we want to do is we want to estimate the number of bacteria per ml in this tube. To estimate the number of bacteria per ml in this tube, we're going to do a standard plate count. The first step of a standard plate count is called a serial dilution. The goal of a serial dilution then is to take some of the bacteria in here, a sample, add it to water here and dilute it again, dilute it again, dilute it again, all the way down through these 7 tubes until we get approximately between 30 and 300 bacteria in a single tube, okay, or in 200 microliters of a single tube. We will then take 200 microliters from that tube, place it on a bacteriological plate. We will then spread the bacteria out across the plate, incubate them, and count the number of colonies. Each colony then is derived from a single bacterium. That single bacterium grew into a colony, and each colony is called a CFU, or colony forming unit. So to begin with, what I want to do is I'm going to take my water here, and I'm going to put 900 microliters of water into each of these tubes. So here's my pipetter set to 900. We'll put 900 into each of these tubes. Since this is water and all of these tubes are sterile, I'm just going to use a single pipetter to put it into all of these tubes rather than changing my pipette tip. All right, now I've just finished adding 900 microliters to each of these tubes of water. We'll get rid of our pipette tip. Our next step then is to mix up our E. coli culture. So I'm just gonna swirl that around until all of our E. coli is evenly distributed throughout that tube. I then want to add 100 microliters of our E. coli culture to our first tube. So to do that, then I've set this pipetter to 100 microliters. I'll get a pipette tip. We will get our E. coli culture that we just mixed up. Keep that lid in my fingers. We will go ahead and flame the tip. Take this down. I'm going to mix it up a little bit by pumping up and down on that gently. And now I have 100 microliters. I'm going to put that 100 microliters into the tube. And then I'm going to get rid of my tip. We'll flame and return the cap. Okay, now what I want to do is I want to mix this up. So I'm going to go in here, I'm going to finger flick this until it's all mixed up. Okay. And then what I'm going to do is I'm going to take 100 microliters from this tube, put it into that tube, mix it, put 100 microliters from this tube into that tube and mix it, so on and so forth until I finish all of those tubes. Okay, so let me just finger flick this very last tube, and we are finished in doing the serial dilution in all of those tubes. Now if we look at the dilution factor, this one is 10 to the negative 1. This one is 1/10 fewer bacteria, 10 to the negative 2, 10 to the negative 3, 10 to the negative 4, 10 to the negative 5, 10 to the negative 6, and 10 to the negative 7. All right, let's move a couple things out of the way here. And I'm gonna put in some plates. We'll remove this, and we'll keep my loop right here. I'm gonna straighten my loop in preparation of the next step. So the next step what I wanna do is I basically wanna take these plates, and you see these plates have already been labeled. This plate is labeled for the section number, the group number. It is 10 to the negative 4, and it is E. coli. So here's my 10 to the negative 4 tube. So I'm going to take 100 microliters, put that 100 microliters onto this plate, 10 to the negative 4. I'll then put 100 microliters from 10 to the negative 5 on our 10 to the negative 5 plate, 100 on our 10 to the negative 6 plate, and 100 on our 10 to the negative 7 plate. Okay, I'll just demonstrate once with the 10 to the negative fourth plate so that you can see that, and then we'll finish up the rest. So what I want to do then is I want my 10 to the negative fourth tube. I don't want to make a mistake there. We'll pull that out. I'm then going to get a pipette tip. I want to take my tube, we'll finger flick the tube really fast. And we'll open this up. And I have this set on 100 microliters. I'll mix it a few times nice and gently. We get 100 microliters in our pipetter. I'm just going to lift this lid up a little bit, and I'm going to put all of that on the plate. Okay, and then we'll get rid of the pipette tip. And now what I want to do is I want to take my loop. I've straightened it out, and you see we have a nice loop at the tip there. So that loop's in good shape. I'll move a couple things out of the way. I will flame the loop going to the top of the cone, downward motion where I get a nice arc on that flame. Now my flame is hot, so I want to be careful. In this case, I have a lot of solution in there, so I'll just go in, gently touch to the solution. Now I want to get an even distribution of bacteria on this plate. So I'm just going to spin that with my left hand while I go back and forth with the loop across the plate, trying to get a really nice even distribution of bacteria across this plate. There are many ways to do this. You can do this with sterile beads, which is very good. You can do this with glass spreaders. This loop method works fine, and we can see what the results will be in a few minutes. Okay, so once I've decided that I've gotten a pretty good even distribution across the plate, we'll put the lid back on the plate. You notice I kept the plate within the sterile area here. I'm going to flame my loop again. Then I will do that same procedure for 10 to the negative fifth, 10 to the negative sixth, and 10 to the negative seventh. On these plates, put them in the incubator and incubate them for 48 hours. Okay, so we've now had 48 hours pass in the blink of an eye. And we're now ready to look at the results. So we have our 4 plates here. Here's our 10 to the negative fourth. We can see a lot of bacteria on here over the plate, definitely more than 300 colonies. Here's our 10 to the negative fifth. This might be the one that we have to count, right? Here's our 10 to the negative sixth and our 10 to the negative seventh. So out of those 4 plates, what we're looking for is trying to count the plate that has between 30 and 300 colonies, okay? And so you as a student making this lab up, you have to figure out which plate to count, and then you want to count that plate. So we'll do a close-up of each plate so that you can go through and make those counts. Each of those dots is a colony. Each colony then is made from 1 bacteria on the plate growing out into the whole colony. That colony is called a CFU, or colony forming unit. The next step in this lab is to perform a turbidimetric analysis. So I have the exact same tube of E. coli here that we use for our standard plate count. Once we're finished with our standard plate count, we're going to know how many bacteria there are per ml in this tube after we incubate it and count our bacteria. Once we know how many bacteria are in this tube, what I'd like to do is set up a standard curve using a spectrophotometer. Spectrophotometer then uses cuvettes. So these are called cuvettes. A spectrophotometer then takes light, shines the light through your solution. If there are any bacteria in here at all, then the light gets stopped by those bacteria and they don't make it onto the other side where there's a phototube. The amount of light that gets stopped is called absorbed. So the spectrophotometer then is going to measure absorbance, and it's going to tell us roughly then, or relatively, how many bacteria there are in the tube. So to start with then, I want to mix this up really well. We'll get in here and get this all mixed up. You can see it mixing up from the bottom because we want an even distribution of bacteria if we're going to get a good estimate in there, okay? Now what I'm going to do is I'm going to put 1.5 mLs into each of these cuvettes. Okay, and we'll just start with that. Put on my pipette tip. We'll then grab the sterile broth, flame the tip of it. Now, since all of these are sterile, I can just use the same pipette tip right now because there is no difference between any of these. During the second step, though, I'm gonna have to change pipette tips for every single procedure because we wanna make sure that we get it right. Okay, I have this set at 750 microliters, so I'm going to have to add another 750 to each one to make a total of 1.5 microliters. All right, that's good. All of these are filled with 1.5 mLs. I'm going to get rid of my pipette tip. This very first tube we're going to use as our blank for our spectrometer. I'm now going to take an empty tube and put it here for our undiluted sample of culture. So we have our culture here. I want to make sure we mix that up again in there. And I want to add 1.5 mLs of this to this first tube, which is going to be our undiluted sample. So we'll just flame the lid here. And I'm just going to mix this a little bit gently just to make sure everything is nice and evenly dispersed in that tube. Okay, and then we're going to put in 1.5 mLs, which is actually 750 microliters. Okay, we'll flame this, do this. Now I have to get rid of my pipette tip every time because there will be different amounts of bacteria in those pipette tips. So I'm on a nice fresh pipette tip. Okay. All right, so now what I'm going to do is I'm going to leave that one set with 1.5 mLs. I'm going to do a second batch where I take this one and I'm going to add the same amount to the second tube. So here's 750 microliters. And now here is 1.5 mLs. So this tube has now been diluted 1 to 2. It has 3 mLs in it. This tube only has 1.5 mLs in it. This is my undiluted sample. So now what I want to do is I want to take this, and we don't have to worry about sterile technique as much because we're not going to be incubating these. And I'm just going to pipette up gently a little bit, and then ultimately take 1.5 mLs or 750. We'll put it in here, and we'll have to do it a second time to make it 1.5 mLs. And I'm going to continue with that same process, 1.5 out of here mixed to here, 1.5 out of here mixed to here. And at the end, then I will have 3 mLs in this and 1.5 mLs in all of those tubes. Okay, so the next step in a turbidimetric analysis is to use the spectrophotometer to determine the absorbance on each of the solutions that we already made. So I'm going to take the spec. We have a firewire connection to our spectrometer. Our USB connection on our computer is down here. So I'm going to plug that in. And we have this spectrometer all ready to go. Now what I want to do is I'm going to go here to Department Applications. We'll go to Logger Pro 3. Open up Logger Pro 3 on our Department Applications. We can see here our x-axis is absorbance. It goes from 0.5 to 1.0 to 1.5 at the top. Our x-axis is wavelength in nanometers. We're going from about 380 nanometers to about 800 nanometers on that scale. Okay, this is visible light. Visible light then goes from purple to blue to green to yellow to orange to red, like the colors of a rainbow. Okay, now let's go ahead and look at our samples here. This is our blank, if you recall, which is straight, pure nutrient or pure broth. Here is our undiluted. Here's our 1 to 2, 1 to 4, 1 to 8, 1 to 16 that we diluted a minute ago. First step is calibration. If we look at our cuvette, you can see that we have an opaque side and a very clean side. So I've gone through and I've taken a Kim wipe and I've wiped all of these off so you can see that these cuvettes are really nice and clean in here. I'm going to put this down to where the clear side goes through, the light goes this direction, and we make sure that we have light running in the correct spot. I'm then going to go up here. I want to go to experiment. I'm going to go down to calibrate, over to spectrometer. And in this case, I need to let it warm up for 87 seconds. I have a little play button, so I'll click play. And what this does is this shows the calibration. So just for fun, we're looking at calibration. Calibration means that it's going to set everything to 0. You notice we have a little bit of trouble here. So if you were measuring wavelengths right here, we probably wouldn't be able to use this spec if it's around 380. However, we're measuring it at 580, so we'll be fine to use the 580, okay? I'm going to hit stop up here. My calibration has worked. I'm now going to take the undiluted sample. Here is our undiluted sample. We'll look at it, make sure that the sides are really clear. There's no scratches or blemishes. Put it in. I'm going to hit play again. For this particular one, I don't care about the original blanking, so I'm going to hit erase and continue. And we have a nice curve there. So we're absorbing here in the purplish range. We're absorbing less and less and less over here. About 580 is right here. So we're wanting to read our number right about there, which is a pretty good number for this particular curve. I'm going to hit stop. Then I'm going to take my sample and put in my 1 to 2. That was undiluted that we used first. Our 1 to 2 is what we're going to use second. I now am clicking start. In this case, I'm going to hit store latest run because I just want to keep that on there just for fun because it's visually cool. And there we go. We can see really nice. This is a 1 to 2. 580 is right here. So we've gone from about here to about here. The higher this number is, the more bacteria are in the sample. I'll hit stop. We'll then switch this out and put in our next one. Make sure that it's nice and clean. We'll hit play again. Store the latest run because it's fun. And we can see again it drops. So we went here to here. This dropped less than this because it's going to be a logarithmic function in there. And so it just continues to drop less and less. All right, we will hit stop again. Take this one out. Put this one in. It's nice and clean. We'll hit play, store latest run. Nice, reduced again. So biggest reduction, less and less. So as we're going in, we can see that reducing even more. And finally, we will hit our last one. And we will hit play and store latest run. And we can see this one's kind of bottomed out here. So it's kind of even right there, just a teeny bit down. Now, we want to take our values, right? All right, so now we can actually look at the data. So here we have our graph. We have our 4 different lines. Absorbance, the higher the absorbance, the more bacteria cells we have in our culture. We're trying to look at 686, so somewhere around here is the absorbance that we want. We want to just go down here, look at this scale for the absorbance, for the exact numbers that we want. And so we just scroll up here, down here until we find 686. And right here is 686. So that's the value you want to record for the purple line. I'm then going to move over. The next one's actually the red line up here. 686 is right there for the red line. Our blue line here, 686 is right there for our blue line. There's our green line. And there is our, what is that, orangish line. And those are the data that you want to record. Okay, so the last part of the lab. Here's an unknown. This unknown, we don't know how many bacteria per ml are in here, but we want to figure out how many bacteria per ml are in here. Now, I'm not going to tell you anything. You have all the results from the last lab. I'm just going to run the spectrometer and figure out how many bacteria per ml are in it. So what I did was I took this unknown and I placed 1.5 mLs into this cuvette. This cuvette has my blank broth. And so now I'm gonna put it way over there. All right, so I'm gonna take my sterile broth, put it in, we're going to calibrate it. We'll go up here, we'll go to experiment, we will calibrate spectrometer. I just used it, so we'll skip the warm up, we'll finish calibration. And press OK, just for fun. I always like to click it to make sure that our calibration is correct. And we can see again a little noise here, but everything is zeroed out. Now in this case, I'm not going to change anything. I'm just going to take this off and put in our unknown culture. Undiluted unknown culture. Here is our readings. We probably want to go to 686 on here, because that value may be important. Hopefully it's important. And here is our 686 right there. And now your job is to figure out how to calculate how many bacteria per ml with that reading.